Hello, can somebody confirm that I can hear you?
Yes, sir.
Thank you. Where we begin, let me inform you that I'm taking some medication for some medical procedures tomorrow, and I'm not handling the medication too well. So I may need to stop the meeting today and go use the bathroom, but hopefully it'll be very brief interruption if an interruption does occur. Are there any questions about that? Somebody's using chat, and let me remind you that when I'm sharing my screen, I cannot see your chat messages, so I do not use chat. You need to unmute your microphone and ask a question if you want to ask me a question. All right, today is April 15th, tax day, and we are supposed to be finishing chapter one and beginning chapter two today. I'm not sure we'll start chapter two today, but I'm going to try. And we have a lot in chapter one to finish. Let me remind you that you have a practice quiz, quiz 00, this Wednesday. It'll cover chapter one, lab 0, and lab 1. Any questions about that? So that's the regular time. You'll have a quiz uh, due at 11:59 p.m. Wednesday night. You do need Respondus Lockdown Browser and Respondus Monitor to take the practice quiz. You also have a plagiarism quiz, which is due 11:59 p.m. on Saturday for April 19th, and that will be covering the plagiarism tutorial. You must achieve 100% on the plagiarism quiz in order to achieve points on your infectious disease project. The infectious disease project is worth 100 points, so you need to get 100% on the plagiarism quiz. On the plagiarism quiz, I rarely have a student who needs to take it more than 2 times. You can take that quiz, I think you can take it up to 7 times. It's the same questions. So if you get a question wrong, you just have to figure out what the answer is and then change your answer. But you can take the plagiarism quiz up to 7 times. Any question about any of that? You do need Respondus Lockdown Browser and Respondus Monitor to take the plagiarism quiz. Let me see if I can unhook this and show you what you need to do to take Respondus Monitor. A little longer than I was hoping. Yeah, that came out. That came out. I think that's it. So if your camera is in a browser, hopefully you can see this browser. I may have to be in the picture before you can see it. Ah, oh, I can't see them on here. Let me see if I can shut down the background. I might not be able to do that from here. Now you can see the monitor on my lap here. What you want to do is, yep, let me shut that on. Are you seeing this now? Or am I not sharing my screen?
I can see
Able
you.
to see it.
You can't see me. Okay. So uh, take your monitor if you need to, and then tilt it down so you can show me where the monitor is sitting, and then move the monitor to show me the right, and then move the monitor to show me the left. If you're not sure what to do, just move to the right and then go up and then down and then go to the left side and show me up and down, and then I'll see the entire background. But I do need to see where the monitor is sitting, and to the right of that and to the left of it. Somebody had a computer and they were very good, and they just went voomp, voomp, and I really didn't see. You need to go really hard with the uh, webcam to show me the background right here, which I'm not sure I'm even seeing that because I'm not seeing the screen now. It's down so far. And then they went whump and whump. And especially if you do it with your hand and you go like that, I'm not really seeing much of the background. You need to go sharp and then go to the left and the right. And actually, all I'm seeing here is the monitor, so I got to go sharper, something like that. If you do not show me your background information, then I have to watch your entire quiz. Let me start the background again. Oh, there it is, video. Which one did I have here? I think I have the Clark College one. There we go. If you don't show me your complete background, then if you ever take your eyes off of the computer screen, like right now I'm looking at the computer monitor screen, if you take your eyes off the monitor, especially if you look down to the left or to the right, I'm going to assume that you've got something to cheat notes on over here. And then if you take your eyes off the monitor, I'm just going to give you a 0 for that quiz because you took your eyes off the monitor and I can't see what you're looking at. So you're getting a 0 on that quiz. And if you don't like that, you can also take the quiz at Clark College. You just need to let me know, and then I will give you the quiz, print it out, and you'll take it in class at Clark College. The room number will be determined um, at the regular time, which would be 5 o'clock on Wednesday night. And if you're not near Clark College, you can schedule to take the exam proctored at another college. You've got to schedule this yourself. Let me know in advance so I can print out the exam and then send them to them. And hopefully you'll do that for the rest of the term. And I will simply need to know in advance, and then I can print out the exam and send it to them. And I do need to know, gosh, about at least 3 days in advance so that I can get it to them in time. Although, if you set it up and I email it to them, I can probably get it to them in about a day. Any questions about any of that?
It's required to be taken at this time.
Okay, you're broke up, so can you repeat that and let me try and listen to that again?
Sorry, I have poor signal. Are the tests required to be taken out of specific?
At a specific what? All right.
Sorry, I've got really bad
I'm
signal
going to
out
stop
here.
sharing my screen and then you can ask your question in a text message, a chat's what it's called. But I got to stop sharing the screen here in order to see the chat message. Am I sharing my screen now? Can somebody confirm, am I not sharing my screen? I don't think I'm sharing my screen, so I'm gonna get out of here. All right, all right, use the chat to give me your question. Are the quizzes required to be taken at a specific time? Yeah, they have to be taken sometime between midnight Tuesday night and 11:59 p.m. Wednesday night. They must be taken at that time. Is that your question? All right, I'm not hearing anything, so I'm going to assume that was your question. How do I get out of chat here? And then restart sharing my screen. All right, are there any other questions? Don't know how that got changed. So let's begin chapter 1. Good, it's showing me where I left off last time. Is this where I left off last time? That's not right. Let me look where 2 here is. I think it's here. This is where we left off last time. Microbiology and microorganisms. And also might be a brief history of microbiology. I think this is where we left off. Is that correct? All right, I'm
It
going
was
to start.
I think it was the other one.
You think it was the other one? All right, so we discussed what comes under the umbrella of microbiology. Uh, a brief history of microbiology largely concerns the debate over life spontaneously generates or the theory of biogenesis that life comes from life. The history of microbiology also concerns the golden age of microbiology. You don't need to know the times of the golden age of microbiology, but it was early microbiology from about 1857 to 1914, when wherever microbiologists look, they were looking at something brand new that nobody had ever seen before. So they were making new discoveries almost all the time in the golden age of microbiology. And this is giving you a timeline or the golden age of microbiology, events before microbiology actually began, like the discovery of the microscope, and then the golden age of microbiology, and then the history of microbiology, which we will briefly cover. History of microbiology, I'm going to state the events in the history of microbiology do giving you a timeline. You do not need to know any of the times except for the very first event, and that is 3.5 billion years ago. The ancestors of bacteria were first seen in the fossil record, where we can see fossils and rocks of the earth. And this is actually showing you. Let me see if I can blow this up. That's an actual fossil of a microorganism. And there's the artist's rendition of what they think they're seeing, which were the ancestors of bacteria and the archaea that we see in the fossil records of Earth greater than 3.5 billion years ago. The next event in the history of microbiology was the Babylonians. You don't need to know the time, but about 4,300 BC wrote down recipes for making beer. And they didn't know this, but you need microorganisms to make beer. And then 600 AD, the Mayans wrote down recipes for making a fermented beverage from cacao that we now call chocolate. And you kind of need to know the events, like before the Mayans wrote down their written record for making cacao. The Babylonians had to write down their written records for making beer. And then before that, we needed to have the first fossil records of microorganisms in the history of Earth. So you just need to know that the fossil record was first, the Babylonians were second, the Mayans were third. You don't need to know the dates of these events. Any question about any of that? All right, the next important event in the history of microbiology was the development of the compound microscope. Janssen developed the microscope. You don't need to know the date, but in 1590, and that had to be done before anyone could look at microbes and see them with a microscope. Robert Hooke was the next person in the history of microbiology. And you don't need to know the date, but in 1665, he's the first microbiologist who took a microscope and looked at living things. And he looked at plant cells and he found little boxes or cells in the plants. And he called these little boxes cells, marking the beginning of the cell theory, which a whole bunch of scientists developed over time. But basically the cell theory is that all living things are composed of cells. And it first started with Robert Hooke looking at plants and seeing little boxes in the plant cells that we called cells. And the cell theory is simply that all living things are composed of cells. It was actually wrote up and written later by Virchow. We'll talk about it later, where he proposed the theory that all living things are composed of cells. But it developed over time as people started looking at different things and they looked at anything that was alive and eventually they saw cells in them. Robert Hooke did look at animal tissue, but he did not see cells in animal tissue. And that is because plant cells have something which is very thick and could be seen in the blurry microscopes that Robert Hooke was looking at. And the primitive microscopes were blurry, and you could not see animal cells in these blurry microscopes because of one thing that they lack and plant cells have. What is that? What do animal cells lack and plant cells have? And that's why Robert Hooke was able to see the cells in plants. Anyone know? You should know that from biology 160 or your earlier microbiology or your, what do you call that? The class you had to take before you could take microbiology. I'm not hearing anything. So what plants have and animals lack is a cell wall. The cell wall is very thick and the primitive microscope could see the cell walls. And then Robert Hooke could see the cells in plant tissue. He could not see the cells in animal tissue because they lack a cell wall. All they have is cell membranes, and the microscopes were too blurry to make out cells in animal tissue, at least back when Robert Hooke was looking around 1665. The above is not specifically microbiology, even Robert Hooke looking at plant cells. This is not microbes. Plants are a organism that is larger than a microorganism. What Robert Hooke did when he was looking at the first plants that he looked at was actually the cork which is the bark of the cork tree. And cork is also used to describe the cork tree itself, as well as the bark of that tree. And then cork is also used what we cap a bottle, and we call it a cork. So that's three ways the term cork is used. And that cap is made out of the bark of the cork tree and why it's called cork. Anyways, when he took a look, let me stop the screen here, at plant cells, what he saw was this. Trying to make straight lines here. Sort of straight. Not going quite high enough there to get it at the top either, but hopefully you can figure this out. And put your last one here. Anyways, in the monastery, which we're going to draw here, so this would be a hallway in the monastery. And I guess this would be, let me erase that right here. Right there. And now I need to get the brush back. So this would be the monastery right here. And the hallway would probably go around like that. Um, Anyways, in the monastery where the monks lived, they had an individual room and it was shaped like this, and they called it a cell. And Robert Hooke saw these plant boxes in plant cells, and it reminded him of the cells of the monk's room. So we coined the term cell after the monk's specific room in the monastery that they called the cell. And that's where the term cell comes from. Anyone got a question about any of that? And that was simply from the shavings of the bark of the cork tree. All right, so in the history of microbiology, it largely composed the hypothesis about how life originates. Originally, there were two competing hypotheses to explain how life began. There was the theory of spontaneous generation, that life arises from a vital force coming into inorganic matter and spontaneously generating life. So that was the theory of spontaneous generation. And then there's the theory of biogenesis, that all life originates from earlier life. Like people say, a baby arises when the two parents come together, and then the sperm fertilizes the egg cell, forming the baby. So the question about where babies came, everyone would agree that they came through biogenesis, that the baby only came from the two parents. But there were other instances where life seemed to spontaneously generate. That says a century ago, they would throw a log into the fire, and sometimes a newt would arise and crawl out of the fire. And scientists would say, or primitive scientists would say, we put the wood into the fire. The two came together, forming the newt. And then the newt crawled out of the fire. In reality, the newt was just in the bark of the tree, and somebody threw the wood into the tree, and the newt felt the heat, and so the newt would crawl out of the fire. But they also saw flies emerging from manure, and they said the vital force came into the manure, the organic matter gave rise to the flies, and then the flies came out of the manure. And then they also saw maggots in decaying corpses or decaying organic living tissue. And they would say the vital force came into the organic matter and gave rise spontaneously to the maggots. Most of these were logical, but they were not scientific. Some were even amusing. Somebody wrote up that if you put sweaty underwear in a jar and put wheat kernels in the bottom of the jar, it would spontaneously give rise to mice. In reality, what had happened was the mice crawled into the jar to get uh, access to the wheat kernels, and then the mice got trapped in the jar and could not climb out of the glass of the jar. And so that's how the mice got into the jar with the sweaty underwear. And the sweaty underwear actually had nothing to do with the mice coming into the jar. Any questions about any of that? All right. Well, one thing about microbes, and that is in the early microbiology, if you made media and then you didn't cap it, with time, the media, which looked like this, would become turbid. And then they would look at things in that media and they'd see microbes growing. And so people said, microbes spontaneously generated in the media. And this almost always happened whenever we made media over time. Even if you capped it over time, it would become turbid. And they would say the vital force came in, giving rise spontaneously to microbes. The first scientific evidence to disprove the theory of spontaneous generation happened by Francesco Redi, and you should know this person. He actually did this in 1668. You don't need to know the date, but you should know that Francesco Redi did the first experiment to disprove spontaneous generation, and actually this was the first documented controlled scientific experiment. So you should know this as the first controlled scientific experiment that was ever written up. Francesco Redi, he got 3 jars and sealed the jars, putting, before he sealed the jars, meat in the jar. And then he got 3 open jars and leave them open. And the maggots appeared in the meat in the open jar, but not in the sealed jar. And the spontaneous generation people said, no, Francesco Redi, you sealed the jar so the vital force could not get in the meat and then give rise to the maggots. So your experiment didn't work, they said. And Francesco Redi very ingeniously thought of a way to do this, and that is he got a jar, put the meat in the jar, and then covered the jar with a fine wire net, and no maggots came into the meat. In reality, what happened was the flies from the open jar landed on the meat, laid their eggs in the meat, which gave rise to the maggots. The flies could not get into the sealed jar or the jar covered with a fine wire net, and so they got no maggots. The point is, after Francesco Redi's day, people would say that complex organisms like maggots or anything is not a microorganism could not spontaneously generate. However, if you use the fine wire netting, microorganisms could get in and then they would grow in the meat or they would grow in the media. And so people still argued that microbes could spontaneously generate after Francesco Redi's experiment. Any question about any of that? In this time, we also have Antony van Leeuwenhoek. I'm not Dutch, so I could be mispronouncing his name. He observed the first microbes using the microscope to actually look at microorganisms. And essentially, wherever he looked, he found microorganisms. He described microorganisms in scrapings from his teeth, in rainwater, in peppercorn infusions, meaning he took peppercorn, put it in water, and let that grow, and he saw microorganisms. And then he saw microorganisms in the water from the River Thames. And the people of London didn't really like that because they were using that water to drink for their drinking water. Any question about any of that? Here is Antony van Leeuwenhoek. He saw cells that were so small that they were not seen by other people for nearly a century. And here are the drawings here. There is a rod bacteria. Here's more rod bacteria. Here is a spirillum. And there, figure E, those are actually cocci. He was seeing cells so small that other microbiologists were not able to see them for nearly a century later. And that's because he was the best microscope builder and the best microscope lens grinder. He actually ground his own lenses, and he was the best lens grinder. And here's a microscope that he built. It's a simple microscope, only using one lens to magnify the specimen, but it was the best microscope of his day because he grinded the lens the best, and he was able to see really small objects clearly, whereas the compound microscopes that were being used were not, the lenses were not ground as finely, and then they were blurry when they put them together, and so people could not see the bacteria, which Antony van Leeuwenhoek actually saw a century earlier than other people. Now, other people, if they used the microscopes he built, could actually see this figure, but any other microscope, you could not see these bacteria or archaea because the microscopes needed 100 years development before they got good enough to actually see these very tiny cells. Any question about any of that? That's actually the primitive microscope. That pen was what he actually stuck things on, and then he is using the primitive microscope there. Let me see if I can blow that up. He's looking at it there. He actually shined that primitive microscope up to the light, usually the sun, to view the object that he was looking at. Any question about any of that? All right. In this time, Rudolf Virchow actually wrote up the cell theory that all living cells arise from pre-existing cells. This became known as the concept of biogenesis. However, arguments still continue. You don't need to know the date here, that microorganisms could still spontaneous generate. And that argument continued until 1861. You don't need to know that date either. But it's a few years after Rudolf Virchow proposed his theory that all cells came from pre-existing cells. And what happened was Louis Pasteur came on the stage and he disproved the spontaneous generation theory and therefore confirmed the theory of biogenesis by his ingenious, famous swan neck flask experiment, where before that, Louis Pasteur also showed that microorganisms are present in the air and that if the air simply came in contact with the media, that the microorganisms would grow in the media. And then also that microbes could be in the tube And so if you put the media in the tube and then seal the tube, there's microorganisms in the tube and they'll give rise to the other microbes in the tube. And he did show, Louis Pasteur, that if you heated the tube and then sealed it, that microorganisms would not grow in that heated tube and then sealed tube. And so what he did was he took a long container with a long neck and put the media in it, and then he heated the media up, and then he heated the tube to make it a swan neck, meaning in this bent shape like that. He showed that no microorganisms would grow in the tube. And then he further showed that you needed to get the media down into this region of the tube before, and then bring the media back into the tube before microorganisms would grow in the tube. And he proposed that microorganisms from the air came into the tube and fell down into the lower portion of the tube, but the microorganisms could not come up the tube against gravity and then come into the media. And that was his swan neck experiment, that the media stayed sterile in the tube that was sterilized, and the swan neck would be open so the vital force could come in into the media, but the vital force did not give rise to the life because the life came from pre-existing cells and they would be stuck right here in the bottom portion of the swan neck there because of gravity. Any question about any of that? And there was a swan neck like this that actually, he's actually the head of the biology department now. Antonio Roberto and he put it in the window there of the Biology 260 lab. And you can actually see that tube. It's been there for over 10 years now, and it's never grown microbes. Eventually, the media will evaporate and there won't be anything there. But the last I checked, which was probably about 2 or 3 years ago, that flask was still in the window of the media. So you can go past the lab and see it there. So after that, people said, no, microbes do not come from spontaneous generation. They only come from the theory of biogenesis. And this led to a number of discoveries in microbiology, such as the relationship between microbes and disease that became known as the germ theory of disease, that microbes could cause disease. This also led to the concept of immunity, that is, a person could become immune to a microbe, and then that person would be exposed to that germ and they would not become sick. And then that also led to the development of antimicrobial drugs, that we could find an antimicrobial drug to use against a germ and then cure somebody from a disease. Any question about any of that? There were a whole bunch of advancements after Louis Pasteur's discovery that microbes could only come from pre-existing other microbes. Pasteur developed the basic concept of aseptic technique, where if you sterilize something, like a bottle of media, and you cap the media, that tube would stay sterile, meaning aseptic technique. And then Koch developed the technique for streaking for isolation using solid media. He initially streaked it out on a potato, and that would not be the best way to do it, but because it'd be very difficult to keep the potato sterile. But someone in his lab developed nutrient agar, a new solid media, and then Dr. Koch streaked it out on first on gelatin plates, and then they did it on agar plate, nutrient agar with Auger. And Dr. Hess was actually working in the same lab as Dr. Koch. Actually, I don't think he was a doctor. I think that was Mr. Hess. But um, anyways, my point is Dr. Koch's technique was important because streaking for isolation on a solid media, you can then streak your, what do you call it, mixed organisms out on a solid plate, get individual cells. And then if you took an individual isolated colony and then streaked it out a second time to get individual isolated colonies, if you took the cells that went through two colony isolation events, you would most likely have a pure culture, meaning a culture that was a colony that was descended from one cell and therefore would give rise to only one type of cells in the colonies. And like I said, if you do that through two colony isolation events, you could then get a pure culture. That was a very easy way to get a pure culture. Louis Pasteur did develop a way of getting a pure culture before Dr. Koch, but it was a much more difficult way. So today in microbiology, to get a pure culture, we use Dr. Koch's method of streaking for isolated colonies, and we do it two times before we say we have a pure culture. Dr. Pasteur's way was he would take different dilutions and then make 10 replicas of that dilution. And if he got one tube out of 9 tubes that got growth and 9 tubes did not get growth, he correctly assumed that the tube with only one out of 10 tubes having growth in it would be derived from close to one cell. And so that likely was going to be a pure culture. And Dr. Pasteur was correct, but that was a very difficult way of getting a pure culture because you had to make dilutions of your organism and you had to make many different dilutions. And then you had to replicate plate that dilution 10 times. And then you had to find one where you only got one tube out of 10 tubes having growth. And then you could get your pure culture. If ever you didn't get one tube out of 10, then obviously you never got a pure culture. Any question about any of that? All right. A little bit after this time, but close to that time, is when Sergei Winogradsky developed the concept of the biogeochemical cycles, like the nitrogen cycle and the sulfur cycle. And the reason why he was able to see these biogeochemical cycle was he was looking at microorganisms living in their natural habitat where there were many microorganisms living together, and different ones gave rise to, for example, the nitrogen cycle. Other microbiologists at the time were generally studying a pure culture because if you have a mixed culture and then the tube becomes yellow, you don't know which species in the mixed culture gave rise to the yellow culture. Could be one of them, it could be some of them, it could be all of them. You just don't know until you study it in a pure culture. But Sergei Winogradsky, in order to study the nitrogen cycle, had to study many different organisms living together because one would do one step of the cycle, another species would do another step of a cycle, and to see the complete cycle, he had to have all the species there. Any question about any of that? All right. A little after that, Pasteur showed that microorganisms are responsible for fermentation. Alcoholic fermentation is the conversion of sugar, like in the grape juice, and the microorganisms would ferment that sugar to make the alcohol, to make the wine. And you can also ferment the sugars in hops to make the beer. And Louis Pasteur showed that the microorganisms were responsible for that alcoholic fermentation. Louis Pasteur also studied further and found that the beer and the wine could become contaminated by another microorganism, which then further, spoiled the alcohol of the beer, and they converted the alcohol in the wine or the beer into acetic acid, vinegar. And that is actually how vinegar was made. And that is the alcohol is fermented by another microorganism to make the acetic acid in the vinegar. And that would spoil the wine or the beer if the microorganism would then ferment the alcohol in the beer or the wine, making the beer or the wine sour or non-drinkable. Any questions about that? So Louis Pasteur showed that microorganisms did the fermentation, and then microorganisms would then spoil the beer or the wine to make the vinegar. At least that's what wine changes into. I'm not sure what beer changes into, except for spoiled beer. Louis Pasteur then showed that the spoilage bacteria could be killed by heat that was not hot enough to evaporate all the alcohol in the wine. And he called this process pasteurization, where you apply a high heat for a short time, and then he used it to pasteurize the beer so that the beer would not spoil, and then you could keep it and cork it or cap it, and then keep it for at least a year or longer and sell it, and it would stay beer. It's used in a wide variety of foods today that we call pasteurization. It's most commonly used with milk, where we pasteurize the milk, and it prevents the immediate spoilage of milk by killing off the many of the spoilage organisms in the milk, and it also kills off any human pathogens, and that's why we pasteurize the milk. It does not sterilize the milk, though. It does not sterilize the beer either when you pasteurize it, but it does prevent the spoilage organisms from immediately spoiling the milk. But there are microorganisms that can survive the pasteurization process. They are reduced in number, but that's why pasteurized milk does a shelf life, but it is longer than the shelf life of raw milk. And I don't know how long it is. It's something like 2 weeks after you pasteurize the milk. The milk can stay refrigerated for 2 weeks. That's longer than unpasteurized milk, which must be thrown out after about a week. Any question about pasteurization? So it does not sterilize the milk or the food item that you're pasteurizing, but it does reduce the number of microorganisms, and then it removes all human pathogens. So fermentation, I don't discuss it here, but we'll talk about it later. There's two processes for fermenting the food. You can do it very briefly at a very high temperature, not boiling, but under boiling, or you can do it at a mildly high temperature for a longer period of time. We'll talk about that later. And that's known as pasteurization, which is most commonly used with milk. It does not sterilize the milk. The concept of fermentation, that microbes could affect the organic matter, making the alcohol, other microbes could grow in the alcohol and ferment it further to acetic acid, gave rise to the theory, the germ theory, and that is, could microbes have a similar relationship with plants and animals? Microorganisms cause disease. This concept became known as the germ theory of disease, that microorganisms could give rise to disease. This was an important advance in the theory of in microbiology, because originally disease was thought to come from poisonous vapors that would be breathed in by the person. So that the doctors would say, oh, you went walking through a swamp, you breathed in the poisonous vapors of the swamp, and that's why you came down with cholera. In reality, cholera was present in the swamp, you breathed in the cholera, and then the patient came down with the disease cholera because they breathed in the cholera when they went walking through the swamp. So that in reality, the person didn't breathe in poisonous vapors, they breathed in the cholera microbes, which gave disease cholera. Any question about that? So that's the germ theory of the disease, an advancement over the theory that you breathe in the poisonous vapors. In the germ theory of disease, I'm not sure how you say this name. I'm not German, but Ignaz Semmelweis advocated hand washing, and then when people or doctors hand wash, they were able to greatly reduce, if not totally reduce, postpartum or uh, pleural fever on the OB-GYN patients. Dr. Semmelweis was in charge of the OB-GYN department of his hospital, and he showed that if the doctors wash their hands, the pregnant patients were much less likely to come down with these two diseases. The doctor who was the head of the hospital was so insulted by Ignaz Semmelweis advocating that doctors should wash their hands before surgery, as well as giving rise to a lady's birth, that he actually fired Ignaz Semmelweis. And then Joseph Lister, you don't need to know the date, but he used chemical disinfectants to prevent surgical wound, where he used, what is this called? Phenol, treated the patient's skin with phenol, and that dramatically reduced the surgery-related infections. He had to treat the surgical instruments with phenol as well, proving that surgery infections are caused by microbes, and that if you use a chemical disinfectant before the surgery, you dramatically reduce the surgery-related infection. Robert Koch then showed the first microorganism that was shown to give rise to the first disease, and that is, he showed that the Bacillus anthracis, this organism, causes the disease anthrax. What he did was he injected Bacillus anthracis into mice and saw that the mouse came down with anthrax. He then wrote up the experimental steps he showed to prove that Bacillus anthrax causes the disease anthrax, and he called this Koch's postulates, the steps we use to go through to show that a specific organism causes a specific disease. This, or Dr. Koch's work, actually proved the germ theory of disease, that specific microorganisms or germs could cause a disease, and he showed that Bacillus anthrax would cause the disease anthrax. Any question about any of that? And then the steps used to show that a specific microorganism causes the disease became known as Koch's postulates. Any question about any of that? All right, around this time, Edward Jenner developed the first vaccine. He made the first vaccine against smallpox. And what he did was he was listening to some of his patients, and apparently there was a milkmaiden who told him, "Oh, I can't get smallpox because I got cowpox." And everyone who comes down with cowpox is essentially immune to smallpox, meaning they could not get smallpox. And Edward Jenner listened to this patient of his and actually experimented on two boys, injecting cowpox virus into them. And then when smallpox came through the village or the city that the boys were living in, they never came down with smallpox. I'm not sure if he actually subjected the boys to smallpox or not, but probably not. But he actually only had his understanding and his knowledge. He actually did not have proof that cowpox would protect the boys from smallpox. And he also did not know what would happen when he gave the boys the cowpox virus. And that is, they got a very slight illness, usually just one pock mark, which was cowpox, usually in the site that he injected it into the boy. And he did that with his own son. And the first boy, gosh, what was the boy's name? I don't remember who the boy was, but you can look at up online, but it's not entirely important. But my point is that Edward Jenner was experimenting on two boys, which would be highly frowned upon today, because he did not have proof that what he was doing was safe, would protect the boy against smallpox, and that injecting cowpox was actually injecting a fairly safe disease into the boys. But he was working with children, and apparently at the time, experimenting on children was perfectly okay at the time. Any questions about any of that? The concept of a vaccine, though, was not understood what was going on. And so other people were not able to make another vaccine because nobody understood why the cowpox virus was protecting against smallpox. And that understanding did not come about until Louis Pasteur came on the scene. And Louis Pasteur actually showed that the cowpox virus was related to the smallpox virus, and then that when you inject cowpox into a patient, the patient builds up immunity, meaning antibodies to the cowpox, and some of that immunity would cross over and react with smallpox, thereby creating immunity to smallpox. So that understanding did not come about until Pasteur's date. And then with Pasteur's understanding, other people were able to make other vaccines where they could make a vaccine against a disease, and then that vaccine would protect against that disease. Louis Pasteur made a number of different vaccines. The first one he made was actually against a chicken disease. He was doing this for the chicken industry. And let me see if I can remember. You wouldn't know that disease anyways, but there was a disease among chicken, and Louis Pasteur made a vaccine against that. He's most famous. He made a couple of different vaccines. He's most famous for making the disease against rabies. He made a vaccine against rabies. And if you treated the patient with this vaccine, the patient would not get the disease rabies. Any question about any of that? The protection of that vaccine we then call the immunity. The patient would be immune to the disease that the vaccine is protecting against, like with cowpox, it protected against smallpox. With uh, the rabies vaccine, it was protecting against the rabies virus. So that would be immunity to the rabies or the smallpox. Any question about any of that? All right. So about this time came the concept of chemotherapy, which originally was used as the treatment of a disease with a chemical. This was proposed by Paul Ehrlich, that he said we could find a magic bullet, which would be a chemical to protect against a disease. Nowadays, chemotherapy is specifically used as treatment with a chemical against a cancer, but originally chemotherapy meant any disease. And in the broad sense today, it could be used against any disease, but today in the common sense, we use it only against cancer. So we will use chemotherapeutic agents or chemotherapeutic chemicals to protect against an infectious disease. And they can be a synthetic drug or an antibiotic. A synthetic drug is one that a person or a man makes. An antibiotic, strictly defined, is a chemical produced by a living organism, a microbe, either a fungi, a bacteria, or an archaea, that inhibits or kills other microbes. Understand this is a textbook definition of an antibiotic. Antibiotics, as it is commonly used, is any, I guess, chemical that protects against a microorganism. So that penicillin G, which is made by a mold, the penicillium mold, is an antibiotic by the textbook definition and the common use, but I think it's carboxylin or even ampicillin is not an antibiotic because it is not made, carboxylin is made in a lab by a person, so it's not an antibiotic. It is a synthetic chemotherapeutic agent, okay, by the textbook definition. And then ampicillin is made by a microorganism, but then a human chemically modifies the ampicillin to make it ampicillin, to make it the antibiotic. And so it is not an antibiotic by the textbook definition. It is, once again, it's a semi-synthetic, meaning humans modify it. Any question about that? Now, in common usage, carboxylin and ampicillin is an antibiotic. Most clinicians will use the common definition. They'll call ampicillin an antibiotic. So in this term, I will use the common definition. Just realize there is a textbook definition that differs, in which an antibiotic is only a chemical that is made by another living organism, a microbe, to be used against another, preventing another microorganism from growing. Any question about that? Like I said, Paul Ehrlich concept the chemotherapy, that where you could find a magic bullet, a chemical that would prevent or cure an infectious disease. And in 1910, what he did was he just studied a bunch of different chemicals, and at about the 500th chemical that he studied in 1910, he found a chemical that was a magic bullet that could actually cure or treat syphilis. It was a synthetic arsenic drug, salvarsan, that could cure syphilis. And he discovered it, like I said, about the 500th drug in his lab that they studied. And the lab people were so discouraged by studying one chemical after another, finding none that could be used as a cure against, they were studying syphilis, but that they put outside the door of this lab the message, abandon all hope, ye who enter here. Any question about any of that? But in 1910, he actually found a magic bullet that could be used to cure a specific disease. And like I said, it was Salvarsan, which he discovered in 1910. You don't need to know the year. You should just know Paul Ehrlich found the first magic bullet, a chemical that could be used to treat syphilis. The first drug that was actually discovered that could be used against a drug was the bark of the cinchona tree or cinchona tree, depending on either pronunciation is correct, that was used to treat malaria. And about what date do you think the first drug to treat malaria was known to Europe? Anyone have a guess? Come on, just give a guess. Well, I'm not going to give it, so you guys will have to look it up because nobody's giving a guess. We'll move on, and um, so in 1910, Dr. Ehrlich discovered a cure for the disease syphilis. Salvarsan was a chemical that could be used to treat syphilis. In 1928, Alexander Fleming discovered the first antibiotic. It was actually penicillin G. And what he did was he was a lousy microbiologist and he streaked out his plate and there was a microbe that fell down from the air. And then he happened to leave the plate at room temperature and then came back after the weekend and saw the plate on his counter. And he noticed the bacteria he streaked out was growing like this, and there was a contaminant growing like that in the plate. And near the contaminant, he noticed there was a region where the bacteria did not grow, and he correctly surmised or hypothesized that something was being produced by this mold and diffusing out of the plate and preventing the growth of the bacteria. And he was correct. That was actually penicillin G diffusing out of the penicillium mold. He had to have a number of mistakes here in order to get this. First, he had to be a sloppy microbiologist, had to have the contaminant fall on the plate, had to be the right contaminant, penicillium. He had to grow it at room temperature to get both the mold to grow and the bacteria to grow. And then correctly made his observation. He was never able to purify the penicillin G. The only thing he could do in the 1920s would be to really grow up the penicillium mold and then get crude extracts of the media where he could filter out the cells from the media, but he couldn't inject the media into a patient. But he could show that if he put the extract on a plate he streaked, that there would be no growth where he put the extract. The first antibiotic that was actually developed and first clinically used was sulfonamide, a sulfur drug, and it was a man-made chemical. So it's actually not really the textbook definition of an antibiotic. It would be an antibacterial drug or in common usage, an antibiotic. It became available in 1935. Clinicians could use sulfonamide in the clinic. Penicillin G never became available in the clinic until the early 1940s, where they grew up the penicillin G in the media and then purified the penicillin G out of the media and then injected it into the patients. And if there's time, I'll try and talk about the first two patients that were tested with penicillin G. It's kind of interesting, but today I don't have time. We've only got 4 minutes left. And um, anyways, penicillin G became available in World War II and sulfonamide available in 1935. And you could inject the patients with these two antibiotics using the common usage of the term there and uh, cure the patient of their diseases if the antibiotic did work against that disease. A penicillin G would only work against most gram positives, wouldn't work against the gram negative. And then sulfonamide, I don't know what it works against. I'd have to look it up. Any question about any of that? All right. Bacteria was once classified as plants, hence the term flora to describe microbes. I learned the term microflora, and if ever I use that term, I'm sorry, that's because that's the term that was around when I took microbiology way back in the ancient times. It's now been replaced by microbiota, and you should use that term instead of microflora. All organisms have a normal microbiota present on all plants and animals that we call their normal microbiota. Usually, these normal microbiota live in harmony with the host. All animals and plants have a symbiotic microbial community living with them, and actually, it's beneficial to them and it's central to their welfare. Any questions about that? There is a human normal microbiota, and then like a cat normal microbiota, and then the bean plant normal microbiota. These are the normal organisms that are living with the host. Like Staphylococcus epidermidis is a microbe that lives on both humans and on cats. I don't know if it lives on beans, but it might. So it'd be the normal microbiota, both humans and cats. But then Bartonella henselae is a normal microbiota of cats because it lives on the cats, doesn't cause them disease. But in Bartonella henselae, if it's growing in a human, it will cause disease in the human. So it's not a part of the normal microbiota of humans. And if you want to know, it actually causes cat scratch fever or cat scratch disease, Bartonella henselae. So the normal microbiota are normally found only in and on the skin, the oral nasal cavities, the upper respiratory tract, the digestive tract, and the urethra. It is usually not found internally. Internal organs, the bones, the muscles, like the spinal tissue. Any question about that? The normal microbiota is actually important at preventing the growth of pathogens. The normal microbiota can compete for resources, for space. The normal microbiota can even secrete toxins that inhibit the growth of the pathogen, like the normal microbiota of our urethra and of our mouth, make the environment more acidic that can prevent the growth of pathogens. Actually, the normal microbiota of our skin make our skin a little more acidic than the skin is normally. The normal microbiota can produce toxins that inhibit the growth of a pathogen. We'll talk a little bit about that later. Like E. coli can secrete a toxin which can prevent the growth of Shigella. We'll talk a little bit about that later. The normal microbiota is important for producing nutrients and growth factors for the patient, like E. coli and everybody's gut helps us digest our food. And then other microorganisms are important in producing folic acid and vitamin K. We get a significant portion of our folic acid and vitamin K from the normal microbiota in our intestines or our colon. Oh, and there it is right there. The normal microbiota are important in our digestion. Any question about any of that? All right. I only have like 3 more slides here, but I'm going to stop now. And we're going to continue with the lab at 6:32. So you have 10-minute break between now and the lab. Any questions before I log off? All right. I will say goodbye. See you in the lab at 10 minutes from now at 6:30.